All right, so today we're going to start, or actually we're going to continue eukaryotic cells, and we're going to do namely the endomembrane system. So we're going to call this Eukaryotic Cells Part 2. On YouTube, it'll be lesson number 8, Part 2. And the main thing that I want to discuss with you guys today is the endomembrane system. Now, about 3 years ago, when I taught this, I told students, but in not a very emphatic way, just to kind of to the point when I said the mitochondria and the chloroplasts are not part of the endomembrane system. Period. That's it. And so they took the test for Unit 2 on cells and diffusion in the plasma membrane, and a disturbing amount of students put that the mitochondria and the chloroplasts are part of the endomembrane system. I'm thinking, is it something I said? Did I say it wrong? What did I do? Why would so many students think that? And so the next year, 2 years ago, I remember jumping up on a desk, banging my hand on the ceiling, pounding my feet on the desk. Everybody looked at me, and all the kids like, okay, he's on heroin. And I said, everybody repeat after me. Okay. The mitochondria and the chloroplasts are not part of the endomembrane system. They all said that back. Like, are we clear? I said, yes, we're clear. Please get your hand off the table. You're freaking us out. I graded the test, and there's still about 3 or 4 students that wrote 2 organelles that are part of the endomembrane system are the mitochondria and the chloroplasts. Like, where is this happening? Where's the disconnect? So I asked them to come back to talk to me, and I said, guys, this has happened for several years in a row. Tell me why. Let me into your brains. Let me into your mind. Why would you write the endomembrane system includes the mitochondria and the chloroplasts? You remember when I stood on the table and acted like a fool? I said, yeah. So what did I say when I did that? The mitochondria and the chloroplasts are not part of the endomembrane system. Good. Why did you write it on your test as if they are? And they said, these are the words, I got the words endomembrane and endosymbiotic confused. They're on flip-flops. Okay, that's obviously probable. Is it avoidable? You better believe it. So these organelles are nothing more than a transport system. They're all originating from the infolding period, where the cell invaginates and it forms a little compartment, and that compartment is going to be a new organelle. Speaking of which, somebody texted me last night saying that the video from yesterday's class was nothing but a black screen. Apparently, I didn't share the actual image of my computer monitor, so I redid the video, and it is on YouTube now. So if you ran into that problem, it is fixed. So these organelles are nothing more than transport. If I wanted this to get all the way to the back of the room, it would go to the endoplasmic reticulum. And then it would go to a vesicle. And then to the Golgi apparatus. And then to another vesicle. And then to the plasma membrane. The plasma membrane spits it out. That is what we're going to do for the bulk of today. We're going to basically make a roster of all the organelles that are part of this endomembrane system and what their roles are. Okay? So, let's get to it. First off, what is the endomembrane system? All the organelles of the endomembrane system can exchange membrane with each other so they can pass on macromolecules. Now, what are some examples of macromolecules? And as I told you, as we get further away from Unit 1, it's going to be harder for you all to remember that. So I want to bring up opportunities for us to jog our memories on Unit 1 biochemistry. What are some examples of macromolecules? Carbs, proteins, and lipids. You're not going to really transfer nucleic acids, just these three. Good. Now, next point I want to make is how in the world can all these different organelles pass this on? This is like having the same currency. You could go to Japan, Norway, China, Mexico, France, Brazil, all these places. You don't have to exchange your money for their money. This is all going to be the same. The reason why is because all their membranes are primarily a phospholipid bilayer. Freddie, I know you've been around the world. You go to China. You've been to China, right? Have you ever been to the Indian Ocean?
Yeah.
Have you ever been to the Pacific Ocean? You've obviously been to the Gulf of Mexico. What if you took a bucket full of water in the Gulf of Mexico and you poured it into the Indian Ocean? Is there a difference? No. They're both salt water. There's not a difference. If you took a membrane from the endoplasmic reticulum and you put it into the membrane of the Golgi apparatus, it's the same thing. So it's just going to blend in, just like taking a bucket of water from the Gulf of Mexico and pouring it into the Indian Ocean. Now, if you took a bucket of pool water, that's different. All the membranes here are phospholipid bilayers for the most part, so they're all basically made up of the same thing. So let's start our roster now, okay? The first one that we are going to do is the rough ER. What does ER stand for? The endoplasmic reticulum. My rule with using initials and acronyms or abbreviations is you can use them so long as you know what they mean. You can't say, what does DNA stand for? DNA. You got to make sure you know. You're allowed to use the initials if you can tell me what they actually mean. So this is the first of many organelles we're going over today. The rough ER is a large sac of membrane that contains pores where ribosomes attach. The reason that this is called the rough ER is that it is covered in ribosomes. It's like a bumpy surface. And when I say ribosome, you say, good, protein. Proteins are produced inside the rough ER by bound ribosomes. There's two types of ribosomes that you can expect to find in a eukaryotic cell. There's bound ribosomes and there's free ribosomes. Bound ribosomes are the ones that are stuck to an ER. Free ribosomes are free floating in the cytoplasm. They're not bound to anything. Okay. So what I'm going to do next is I'm going to try to draw the ER. It's not going to be a pretty picture. It looks like a coral reef. It's just a bunch of squiggles and a bunch of curves. I mean, that's pretty much it. And because it's a rough ER, we're going to cover it in ribosomes. These red dots are ribosomes. And what do ribosomes produce? Right, so that big purple dot right here, that's going to be our protein that we're going to follow all the way through. So because we're following a protein, guys, I almost forgot to mention this. We're going to follow the protein pathway. There's a protein pathway and a lipid pathway. We're going to follow the protein pathway. And we're going to follow the protein pathway to exocytosis. Are we exiting the cell or are we going to enter the cell? Right. All right. So I'm going to zoom in on this little area right here. I'm going to zoom in on that. So let's do a little hashtag zoom in and see what's actually going on. So this blue line represents the membrane of the rough ER. This is inside the organelle. This is outside the organelle. We want to get this protein out of there. So this is going to be like a little storytelling. The organelle is going to form a little bubble. This is called budding. And inside that little bud is a protein, the one that we're following. So you go to the next stage. And we now have our vesicle. The vesicle is like the Tupperware of the cell. It's just meant to move things from point A to point B. Now, I want to remind you, you guys don't have to draw this if you don't want to, but if you were to actually zoom in on what this vesicle looked like, it would look like this. What does that look like to you? That's a phospholipid bilayer, and inside we have our little protein that's hanging out. The endoplasmic reticulum is mostly a phospholipid bilayer, which means this vesicle is mostly a phospholipid bilayer. So let's define what a vesicle is now. Again, this is like the Tupperware of the cell. It's a small sac of membrane used to transport macromolecules from organelle to organelle. Okay. So what we have so far is we have this protein that was formed inside the ER. It's a rough ER. The protein needs to make its way out of the cell. So the protein was pushed to the edge of the ER. It formed a vesicle, and this vesicle is now going to move on to its next stop. The next part is going to be the cytoskeleton. Now, when we watched a video on Monday about the inner life of the cell, do you guys remember that little motor protein that was walking like this? What was that little motor protein carrying? A what? It was carrying a vesicle. What was that little motor protein walking on top of? It was walking on top of a component of the cytoskeleton. Now, this whole vesicle is like Tupperware, or you can say it's a package, something of that effect. Think of Amazon for a second. Does the company of Amazon own the distribution warehouses that they build? Yes. Does the company of Amazon own the Amazon vans? Yes. Does the company of Amazon pay the people that deliver the packages? Yes. Does the company of Amazon own the roads that they drive their vans on? No, they do not. The cytoskeleton is technically not part of the endomembrane system, but it is used by the endomembrane system. Just like FedEx doesn't own Dale Mabry, but they use Dale Mabry to get from point A to point B. So I want to make sure you guys have this disclaimer. It is not part of the endomembrane system. Yes, Reed?
So is the cytoskeleton organelle at all?
No. Not everything in the cell is an organelle. Is everything in your body an organ? Right. Okay, so this one's pretty simple. The cytoskeleton provides a path for the vesicle to travel in. Okay. The cytoskeleton is to the cell what your skeleton is to you. It gives you support, it gives you structure, it gives you shape. Without the cytoskeleton, the cell would probably cave in on itself. Now, let's say we have a bunch of cytoskeletal filaments. We got a little filament going this way, we got a filament going that way, and we got a filament going this way. We're going to draw our little motor protein. This cute little guy. He's got two little feet. And as Grace Mary said, it was carrying a motor protein, or excuse me, it was carrying a vesicle. So let's draw our vesicle. And inside the vesicle is the protein. So this motor protein needs to go the right way to get to the next stop on this tour of the cell in the endomembrane system. And the next stop is going to be the Golgi body, or the Golgi apparatus. The Golgi is sacs of membrane that help proteins fold into their final form. The Golgi is also known as the post office of the cell. It receives packages and repackages them for their final destination. Just like the mitochondria is the powerhouse of the cell, this is the post office of the cell. And it kind of looks like this. It's kind of hard to draw. It's just a series of flattened stacks. And it is made up of the same membrane that everything else in the cell is made of. It's made up of a phospholipid bilayer. So let's say I'm going to zoom in right here, okay? We're going to hashtag zoom in, see what's actually happening on a closer scale. So let's go in further in. Here's our vesicle. Inside of our vesicle is our protein. And it is knocking on the door of the Golgi apparatus. So this is the membrane of the Golgi. This is outside the Golgi, this is inside the Golgi. And by the way, I've been saying the word membrane a lot. I can imagine if I were to walk around this room and say, so-and-so, what's a membrane? You might have trouble trying to determine that. So even if you ask Siri, hey Siri, what is a membrane?
A A membrane is a selective barrier. It allows some things to pass through but stops others. Would you like to hear?
That's it. It's a selective barrier that allows some particles to pass through and not allow others. That's all it is. So let's continue this. Because the vesicle is made up of a phospholipid bilayer, and the Golgi apparatus is made up of a phospholipid bilayer, is the vesicle going to be able to blend into the Golgi? Yes. So it's going to look like this. The vesicle is basically joined with the Golgi, and it's going to become part of the Golgi. And in doing so, you now have the protein inside the Golgi body. Freddie, what happened to the vesicle between the second and third part there? It went to the other side? I don't see it on the other side. I'm talking about the vesicle, not the protein. What happened to the vesicle, the blue part? It disappeared. Well, matter can't just appear and disappear. Anybody tell me what happened to the vesicle? Riley? It became part of the Golgi's membrane because, Riley, what is the vesicle made of? A phospholipid bilayer. What is the Golgi apparatus's membrane made out of? You get it, Freddie? It's just they join together. It's like that example I see. You take water from the Gulf, you pour it in the Indian Ocean. It's not going to change. It's all the same stuff. Okay, so now this protein is going to be inside the Golgi apparatus. It's going to be shaped and structured for its final leg of its journey. That's going to take us to the plasma membrane. Also called the cell membrane, same thing. The plasma membrane is a membrane that separates the outside environment from the inside. So, hashtag zoom in. Let's go in further and see what's actually happening. So, there's going to be a little more here, guys. Here's the other side of the Golgi's membrane. So this is the Golgi. This is inside the Golgi. This is outside the Golgi. And here's our protein, ready to leave. And notice how it looks a little different. The Golgi has twisted it and shaped it to do whatever job that this protein is supposed to do. We would say that the protein is kind of like, it's now mature. It's in an actual protein shape. So, the Golgi is going to form a new vesicle. What is this stage known as? What is that Golgi doing? B word. It's budding. That's right. And we now have a new vesicle with the mature protein inside. And this vesicle is now ready for the final leg of its journey. It's going to head towards the plasma membrane. The plasma membrane is what separates the internal environment itself from the external environment. So this is inside the membrane, this is out. When the vesicle reaches the membrane, it's going to fuse with it. Freddie, why is this vesicle able to fuse with the plasma membrane? Yep. What is the membrane made of? Same thing as the majority of your cell membrane. Phospholipid bilayer. Okay. And now, the protein has been diffused out of the cell. What form of diffusion is this, where you have a big, monstrously sized molecule that you want to get out of the cell? It's called bulk transport, but which kind exactly? It's leaving the cell, exiting the cell. Rosemary? Exocytosis. What is the opposite of exocytosis? If you wanted to do endocytosis, you would do everything that we just did, except you would go in the opposite direction. You would start with the membrane, you would end with the ER. Yeah, sure. Are there any questions with any part of the endomembrane system or any part of the endomembrane system? Yes. Yeah, you'll need to know all the different stops in the proper sequence. But you also need to know, are you doing exocytosis, which means you're leaving, or are you doing endocytosis, which means you're coming in? So if I ask you to identify all the organelles in order for endocytosis, you would start with the plasma membrane, and you would do the reverse of what we just did. If I ask you to trace all the stages of exocytosis, you would start with what we did here, the ER, and then work your way all the way to the membrane. Okay, everybody, let's keep this thing going. The next pathway is going to be significantly shorter. This is going to be called the lipid pathway. The lipid pathway is basically made up of the smooth ER. What does the smooth ER lack that the rough ER has? Ribosomes. It's not bumpy anymore. This is a large sac of membrane that produces plasma membrane and lipids. So the actual composition of the plasma membrane can get a lot of its contributions from the smooth ER. The smooth ER mainly makes lipids. Are lipids hydrophobic or hydrophilic? So are they polar or non-polar? Good. Somebody in first period said about the Golgi, and this was a really great question, we'll share with you. She asked, if the Golgi apparatus makes a bunch of these vesicles, wouldn't the Golgi apparatus run out of membrane? It's like, if I keep spending money, won't I run out of money? Yeah, that's, your logic is sound. So what do you do when it's running out of membrane? Well, smooth ER to the rescue. The smooth ER sends empty vesicles to organelles that need to grow because they've used up a lot of their resources. Also, the smooth ER is involved in detoxification. Whenever you take legal or illegal drugs, alcohol, whatever, and it gets into your bloodstream, your liver is responsible for really cleaning all that junk out. You know, too much aspirin, too much painkiller, that can actually affect your liver function. Obviously, too much alcohol and drugs can affect your liver function. Your liver can regenerate itself, but you don't want to bombard it, because some people get sclerosis of the liver, which means scarred up liver. It's just been beat up and pulverized by all the different poisons, if you will, that have been introduced to the body. And so you would expect a lot of smooth ER inside your hepatocytes. So a hepatocyte is a liver cell. I'm sure you've heard of the disease hepatitis. That means liver disease or inflammation of the liver. Hepa means liver. So hepatocytes are liver cells. Saw a hand go up. What's up? Yeah. Riley? Well, that's why it receives a lot of the vesicles from the plasma membrane. So it's like a system of give and take. But also, you know, you're supposed to get lipids in your diet. You got to have that. Okay, we're actually getting close to wrapping up, guys. The next second to last portion is going to be digestion and detoxification. So there's two parts here. These aren't really organelles. They're more like vesicles. If a vesicle is a Tupperware, a lysosome is Tupperware with battery acid in it. A lysosome is a small sac of membrane that contains hydrolytic enzymes that break large molecules into small molecules. What does this word mean? Hydrolytic. What word do you kind of see with hydrolytic? It's an early school year, so you don't have a lot to choose from. Hydrolysis. Good job, Nikita. What is hydrolysis? Is it an introduction of water or a removal of water? It's the introduction of water. Does it want to break down molecules or build up molecules? All right. So it has enzymes that break down molecules by pretty much adding OH and H. What type of cells in your body do you think have a lot of lysosomes, knowing that their function is to break things down and to basically digest them and destroy them? Someone on this side of the room. Anybody? What type of cells in your body do you think have a lot of lysosomes, knowing the function of lysosomes? Stomach cells secrete acid outside of them. What type of cells would engulf something and digest it inside the cell?
Oh, white blood?
White blood cells, right. We call them WBCs, leukocytes and lymphocytes. These have lots of lysosomes. Not only do lysosomes break down foreign and unwanted material like bacteria and fungi and parasites and viruses, but they'll also break down old, broken down, beat up organelles that you have. It's kind of like you have this old piece of crap bicycle in your garage. It's just taking up space. A lysosome would destroy that. Same thing applies to like an old, beat up piece of crap organelle. It just doesn't work anymore. It's taking up space, destroy it. And that's what lysosomes do. The other one is going to be called a peroxisome. You kind of hear the word peroxide in there. These contain enzymes that detoxify the cell. Now, you guys know that you need water, you need oxygen, you need sugar and carbs to survive. Well, your body produces some byproducts that if they were in a high enough concentration, they would be very deadly for you. Carbon dioxide is a great example of it. If you did not exhale carbon dioxide, it can actually make your blood have a low pH, which is acidic. Well, another byproduct of cell function is hydrogen peroxide, the same stuff that's in those brown bottles you buy at the store. Hydrogen peroxide is H2O2. It's a byproduct of cell respiration. too much of it is very dangerous and potentially deadly to a cell. So you have these peroxisomes, which have enzymes that basically convert H2O2 into water and oxygen. And obviously this is unbalanced, but if I were to balance it, we could get the right ratios. Whenever you have a scab or an open wound and you put some hydrogen peroxide on it, you think it fizzes and you think it's disinfecting it. It's not. Your blood has an enzyme called catalase. And the catalase is what triggers the hydrogen peroxide to be converted to water and oxygen gas. So when it bubbles and fizzes, it might just kind of clean out the wound, which is what water would do. But you're really not destroying anything. You're not destroying bacteria or anything like that. You're just really kind of cleaning it. Yes? Because we think it's actually doing something. It does a little. Like I said, it cleans, but it's not going to kill. You want things like a disinfectant or a fungicide or an antibiotic to try to kill any sort of infection. Okay? And we're up to the last part, folks. Storage organelles. The first one are called vacuoles. You don't see these all too often in an animal cell. They're more plentiful in a plant cell. These are large sacs of membrane for storing food and water. This is going to be long-term storage. You know, you have food vacuoles. They're meant to hold food for a long time. You have water vacuoles, which are meant to hold water for a long time. In fact, with a lot of plant cells, up to 90% of a plant cell, let's say that square is a plant cell, up to 90% of it could be a central vacuole filled with water. If a plant leaf was to look really shriveled and pathetic, it's likely that the vacuoles have no more water in them. So if you water the plant, the water will eventually reach the leaves and the cells. It'll fill the vacuole up and the plant will look nice and healthy again and perky. And then we have one more structure and we're done. And this is basically the center of the cell. It holds the most important stuff of the cell, the nucleus. And the nucleus is covered by a casing called the nuclear envelope.
Please pay attention to the following announcements. Anyone wanting to run
Pausing
for
Zoom,
freshman
hold
or
on.
sophomore class officers this year, be sure to fill out the application located on Canvas and submit it by this Friday. Also, nominations for senior homecoming court are now open on Canvas on the senior class of 2021 page, and they will be accepted until this Friday.
Cool. Okay, the nuclear envelope.
And one more additional announcement for Mr. Bush.
Okay. Hey folks, I just wanted to reach out to congratulate our Excellence in Education Award winners for this year, 2021 school year. Our honor maker, Diversity Educator of the Year is none other than Miss Brandy Litwack in our Guidance Department. Our Instructional Support Employee of the Year is Miss Lisette Solar Campos in the main office. And our 2021 school year Teacher of the Year is Miss Drumroll.
Drumstick.
Christy Rainbow. Congratulations, folks, and please join me if you see them and congratulate them for all of their hard work and dedication to our school. This came about pretty fast, so we know that these people have been consistent throughout the year. That's why they, throughout the years, that's why they've received these wonderful awards here. So again, congratulations to each one of them, and we look forward to great things.
Seniors, you are now dismissed.
Okay, let's finish this off. The nuclear envelope is a double phospholipid bilayer. Sometimes this is a little hard to understand, so I'm going to draw it for you guys. How many phospholipid layers do I have right there? One, not half. No. That's what I said. How many phospholipid layers? That's one. What do we call your membrane? A bilayer. How many is bi? Two. So it takes two layers to make a bilayer. So how many individual layers is a double phospholipid bilayer? A double bilayer. 4. See, I told you, sometimes it's a little rough. Give me 30 seconds and I'll get you guys out of here. This is what the nuclear envelope looks like. It is very thick because it needs to protect the nucleus. There are pores all throughout it. So here's your last description. This is a large sac of membrane that contains pores. They're called nuclear pores that allow certain things in and out. And finally, this is storage. What does the nucleus store? It protects and stores DNA. Okay, next time we will talk about the power organelles.